We were chatting about where the breakthroughs in medicine, or frankly anything, come from. And it's not usually from the research organizations or the mainstream areas of study. So you work as a cardiologist, but you had a friend.
A very close friend who was suffering from Parkinson's disease. And he was starting to dwindle, and he wanted me to come with him to his doctor's appointments to serve as his advocate. And I said, happy to do that. And then he said, and I need you to figure out a way to treat the disease. And so I decided I would start reading so that at least I could ask some good questions. And in the process of doing that, started finding some gaps and some discrepancies in the standard teaching and in the standard practice of how Parkinson's was understood, how Parkinson's was treated. And that eventually led me through a lot of work to identify a different mechanism for the disease than has been reported previously, and identified a drug that could be repurposed and therefore brought into clinical trials quickly to address this abnormality.
Now, 40 plus years of work in Parkinson's, and everybody is dead sure that the disease is a result of a lack of dopamine in the brain.
Exactly. And so what happened was, starting in 1960, when the first report was published, what investigators did is they biopsied a chunk of the brain that had the relevant part that affects movement, that controls movement, and they took that piece of tissue and basically mashed it up. It was a little more elegant than this.
Not much more.
But not much more. And so they were looking at the fluid levels from outside the cells combined with inside the cells. And the cells that they were looking at included what's referred to as the dopaminergic neurons, the brain cells that produce dopamine, that enable movement, along with a whole bunch of other kinds of cells that are in the brain. So in this soup, they made measurements and saw that dopamine concentration was much, much lower than in a similar soup from brains of people who had just died without Parkinson's. So there was this belief that dopamine levels were low. By increasing them or increasing dopamine activity, there was improvement, and that's the right way to do it. And this became important because as I continued to read, and I basically attacked the disease sequentially, so I started reading from 60 and to the future, I got to the point where I learned that the number of neurons is reduced as well. I also got to the point where I realized that the number of these so-called vesicles, which are essentially little balls inside the cell that store the dopamine and help release it to send the nervous system signal forward and allow movement, those were reduced. So I thought, well, if I've got the total amount of dopamine and I know the number of cells and I know the number of vesicles, I can start out with simple algebra and then later go to a meta-analysis and get all formal, but basically figure out what the average amount of dopamine is inside the cell, that's free inside the cell. Nobody had ever measured that. Nobody had ever calculated that. I can't find anybody who's even said, we need to know this. But in essence, we need to know this because if you don't have enough, you can't send a nervous system signal, so you can't move. And if you have too much, it's toxic. So it causes dysfunction of the cells and then kills the cells.
This is the wow moment for me when we were on the phone, when he said, on the contrary, not only isn't it the causation, but it's toxic if it's in too large a dose, and it could actually be causing the problem. This was like, wow. And when you got to that point, it was a wow for you.
It was a wow for me. I thought, wow, this is kind of crazy. How could everything be so backwards? And it was part of the process, realizing that there's all of these situations where we're taught something. There's this standard teaching, there's this source that's revered or a person that's listened to, and yet nobody's looked at the source information. I was fortunate enough when I was in training as a fellow in cardiology to be able to start the first phase 2 trial of a beta blocker in heart failure. Now, when I mention that to people, they'll know what a beta blocker is. And if they're sophisticated medically, they'll know that beta blockers are standard treatment for heart failure. When we started that study, beta blockers were all contraindicated. Basically, we were trying to do beta stimulation. And so I witnessed that flip because I was in the middle of that flip, and the biology is actually surprisingly similar between the two things, is the way it looked to me at this point. Because dopamine toxic in Parkinson's, a related compound called norepinephrine, which is like adrenaline, toxic in heart failure. So I thought, well, I just need to do the same thing simplistically. In Parkinson's, I'll just get a dopamine blocker. So I started reading about dopamine receptors in the brain. Wow, are they more complicated in their distribution and function than beta receptors are in the heart? It's like, well, that's not going to work. And I was looking for an existing drug because repurposing a drug
Because repurposing is easier.
is so much more time and money efficient. Think about it for a second. You've got a drug, especially if it's a repurposed approved drug, you've got a drug where you already know that it's not going to cause liver failure, that it's not going to cause cardiac arrhythmic death. All these crazy things that happen in drug development, that are why these programs had to be so big to look for these rare catastrophic things and take so long. I already know they don't exist with this drug. And this drug has been used generally short term, but for decades without surprising safety stuff.
How did you find that drug? What was the properties and how it behaved that became attractive to you?
Well, again, I'm kind of old-fashioned. I started when beta blockers were contraindicated in heart failure. So, you look at standard pharmacologic strategies to treat a disease. So, everybody's talking about biologics and gene therapies and all that sort of stuff. I'm old-fashioned. I'm a pharmacology kind of person, not trained as a pharmacologist, but that's how I approach these things.
Goes back to being at the Holiday Inn Express last night.
Exactly. And I couldn't use a dopamine blocker because it was too complicated for me to understand how to do it. So I thought, well, I'll take the other approach that's been used forever, which is finding the rate-limiting enzyme for the synthesis of a molecule and blocking that enzyme. So the rate-limiting enzyme for the synthesis of dopamine is something called tyrosine hydroxylase. I thought, okay, I can't do a dopamine blocker. I'll do a tyrosine hydroxylase inhibitor. So it's old-fashioned pharmacology.
Process.
So in addition to identifying the mechanism that nobody's gone after, identifying a drug that most Parkinson's experts' first response to me when I suggested is, you're going to hurt somebody. And then I showed them the data that says, no, I will not hurt somebody and I'll probably improve them. I now have 3 sets of data from 3 preclinical models using the drug, showing that
in animal testing.
In animal testing or cell testing, showing that it reverses cell dysfunction, cell pathology, and restores movement almost to baseline.
Wow.
And so now I'm in the fundraising stage to start a phase 2 clinical trial. I've already engaged with the FDA. I've got basically everything set up where as soon as I get funding, an IND is submitted, and the trial started in 6 months. And within about 6 months of there, we'll start seeing the first clinical data.
How exciting. $5 million. How do you raise that? Come to a conference with a bunch of smart rich people?
I'm looking. I'm looking for investors who allow me to talk to their experts early in the process. I've got a first-class, world-class scientific advisory board. I've got several clinical trial sites ready to go with people who looked at the data with me to establish the mechanism, to understand the pharmacology, to see the effects of the drug in these preclinical models, who are on board saying, look, you're enthusiastic, we've got equipoise, which is what I'm looking for in an investigator, we believe the data support doing a study.
Because there's a lot of people who will really benefit from your work. Thank you.
Thank you.